you very much, Ondra, for agreeing to present your recent work. Really happy to have you here today, and the floor is yours.
Thank you very much, Honzo, for the introduction and for the invitation. I came here today, or I was invited, I should say, to speak about our recent paper that came out in M Systems. And this paper is on the catabolism of acetylseringon and cometabolic transformation of 2,4,6-trichlorophenol by a novel monooxygenase. I will just first check whether everyone can hear me well. Are you guys okay? Can you see the presentation and can you hear me? Well, no one's saying anything because all the mics are muted. So I'll be assuming that everything is fine. So first of all, I will get you into a little bit of context, and I should also mention that I am here together with the first author of the paper, Tomáš Engel. So he's gonna wave to you. Good, thank you, Tomiku. And let me now briefly walk you through the entire story and how we got to it. I should start with okay, so this is it. Okay, good. So I should start with how this research fits in the overall scope of our laboratory. So two of the main topics that we are dealing with in the laboratory, that is called chemoecology of microorganisms lab, is that we're exploring the chemoecology of plant microbial interactions. So we seek to identify how plant metabolites affect the composition of soil microbial communities and how these microorganisms degrade plant metabolites and pollutants. And we also try to identify metabolically active microbes, and we look into their metabolic pathways. To do so, we do stuff such as stable isotope probing, selective enrichments, metagenomics, and or EPIC PCR. So maybe about some of these techniques, I will tell you a little bit more sometime else. But today we're going to be speaking mostly about liquid enrichment cultures and metagenomics. Okay. So in our lab, we have been working for a quite long time under the hypothesis that we call a secondary metabolite hypothesis. And this hypothesis pretty much says or is based on the fact that some of the secondary plant metabolites that you can see here as the structures labeled C, D, and E are very similar to anthropogenic pollutants, such as the one that are labeled A and B. And there are basically two options that would explain this secondary metabolite hypothesis. The first one would be that the bacteria use broad specificity enzymes to degrade secondary metabolites, and by accident, or let's say fortuitously, they can also degrade some anthropogenic pollutants because they are structurally similar. Or the second part of the hypothesis could be that some bacterial enzymes were originally intended for detoxification or degradation of species, and they were sort of, given especially the environmental circumstances, they sort of repurposed to handle the degradation of anthropogenic pollutants. Then we speak about something that we call catalytically promiscuous enzymes. So taking these two things into account, we basically work under the hypothesis that plant secondary metabolites that are released into the soil by rhizodeposits and also by turnover of plant-derived organic matter can act as substrates for biodegradative enzymes, and they can also induce biodegradative genes in soil bacteria. And thereby, these compounds increase the overall contaminant biodegradation potential in the overall soil microbial community. And because this paper is mostly about acetylseringon being a part of lignin, I have to give you some sort of introduction on lignin and the compounds that are derived from lignin. So basically, we can say that the degradation of lignin is a vast source of phenolic species because lignin is a complex plant biopolymer that is mostly composed of phenylpropanoid monomers. I usually, when I speak to students, I say that lignin is based on different kinds of phenols, and that's the major reason why we are linking the degradation lignin to the degradation of species. And Especially at the Institute of Microbiology of Czech Academy of Sciences, their lignin degradation is investigated, and it is mostly the degradation of lignin by fungi. Uh, However, we worked under the hypothesis that also bacteria can contribute to the degradation of lignin, especially by degrading lignin monomers and dimers. Recently, with some of our results, we have been able to see that they also have lignin depolymerization enzymes, but that's another story, a different story that I might be speaking about sometime. As I already stated, we started with the fact that this consortium that originated in the compost soil, and once again, this compost soil is a fascinating and very efficient ecosystem, because what we do is that we feed the compost with grass every week, and then every day it gets some peels from grapefruit, some peels from oranges, from tangerines, from bananas. It also gets a lot of eggshells, and this normal, I would say, bio waste that is produced by a regular household. And in our house is inhabited by 6 people, so we produce a lot of, I would say, organic waste. And all of this waste ends up in this compost, and after half a year, you don't see anything there, just the soil. The soil is dark, it's nice, it smells really nice, and if you plant the plant there, it grows really well and efficiently. So after we enrich the microbial communities or microbial consortia on acetoseringon from this soil, and we fed them with chlorophenols, we saw 100 depletion of 2,4,6-trichlorophenol, and almost 50% depletion of 2,6-dichlorophenol. So we definitely saw that selected chlorophenols are efficiently degraded by this consortium, which was grown on acetoseringon. What was even more interesting was when we tried to look at what enzyme is responsible for this degradation. And using several types of experiments and a lot of endeavor by my PhD student Tomik that you saw at the beginning of the talk, we were able to, or he was able to identify that the enzyme responsible for this degradation is a quite new FAD-dependent monooxygenase. We designated this monooxygenase ASDA. It stands for acetoseringon degradation. And we were able to identify that the gene that encodes for this enzyme is located or harbored on the genome of Pseudomonas rhizophila, which was the major taxon when you saw the composition of the liquid enrichment culture. This Pseudomonas rhizophila was the major taxon that was about 70% in abundance, if I'm not mistaken. We also looked at how this newly identified FAD-dependent monooxygenase is induced by several species or secondary plant metabolites. So you can see that among the compounds tested, we saw that the transcription of the gene that encodes for this monooxygenase is stimulated by the presence of acetoseringon and also orsinol. All of the other compounds were not significantly different from the, I would say, zero transcription, or let's say the transcription without any possible inducer. What was important to us was how this monooxygenase acts on acetoseringon. So we did some tests in which during which we collaborated with the group of Tomáš Seidhaml at the Institute of Microbiology, and we were able to identify that indeed this enzyme acts as a monooxygenase. So you can see here when it transforms acetoseringon, it simply introduces one OH group in the position 2 on the second carbon. And when you take a look at the 2,4,6-trichlorophenol, the enzyme also acts as a monooxygenase, but as I was speaking about the catalytic promiscuity, it also at the same time I would say, cleaves off the chlorine atom. So at the same time, it acts as a dechlorinase. This is not something that would be totally unusual, because especially environmental bacterial dioxygenases are enzymes that normally have multiple enzymatic activities, and they normally act on different kinds of substrates. Sometimes the number of substrates ranges from tens to even hundreds different substrates. The most known enzyme from this point of view is probably the toluene dioxygenase from Pseudomonas butyrate-1. Anyway, so we were able to see that our enzyme has a dual activity. It can act on acetylsyringon. Probably acetylsyringon might be the natural substrate, and 2,4,6-trichlorophenol is probably the co-substrate. In the presence of acetylsyringon, this enzyme or the production of this enzyme is stimulated. And in the presence of acetylsyringon, also 2,4,6-trichlorophenol is transformed to the product that is called 2,6-dichloroquinone. So once again, it is an activity that had been known, but the single and specific enzyme that is responsible for this activity has not been known. So that's one of the major novelties of our paper, that we were able to discover this novel enzyme that is involved in the transformation of 2,4,6-trichlorophenol. We also, because it is, as I already said, it is fairly normal for environmental oxygenases and oxygenases in soil bacteria, that they are capable of breaking down several structures. So we looked at different lignin monomers to see whether some other ones, except for acetylsyringon, can be transformed by this specific enzyme ASDA. So you can see here that there is a lot of compounds that can be actually degraded by the pure culture Pseudomonas rhizophila AS1, which is the bacterium that harbors the ASDA enzyme. But when we looked specifically using genetic engineering tools at which of these compounds are specifically transformed by ASDA, we saw that it is only acetylsyringon and to certain extent vanillin. You can see that the second bars in each couples is only present in the case of acetylsyringon and vanillin. We also wanted to know a little bit more about the ecological relevance of the ASDA gene. So we were first wondering how come that it hadn't been discovered until we came with our research and with our cultures. So we investigated more than 300 different kinds of metagenomes that are available out there, and we searched for those ones that harbored Pseudomonas rhizophila. And we were only able to detect the ASDA gene in 9 of these metagenomes. So with this, we were like, okay, so it looks like it's not a very abundant gene. Then we also investigated the overall metagenomes that are to a certain extent associated with the turnover of plant-derived organic matter. So we found few other ASDA positive hits in plant-associated environments or other rhizosphere environments. Thereby, we got to a conclusion that the gene may be present in microbial populations or soil microbial populations in quite a low abundance. Yet it looks like it is a specialized enzyme for the purposes of the degradation of originally probably acetylsyringon and fortuitously also certain pollutants such as the trichlorophenol. With this, I should somehow sum up the significance of our paper and introduce a little bit the further research that we would like to do on this topic. So I have to highlight that our study unveiled hitherto unrecognized route for the degradation of acetylsyringon, or I should say a transformation route, because we haven't been able to identify the action of all of the enzymes in the operon. However, that's the plan, of course. So we basically identified an enzyme that exhibits dual activity in lignin-derived aromatic catabolism and also environmental pollutant transformation. And as I already said, although the mechanisms underlying TCP degradation had been known, the specific enzyme that is responsible for the activity had been unknown until recently. And that's pretty much the knowledge gap that has now been addressed by the existence of ASDA as the enzyme that we designated by this short ASDA. Anyway, so what's next? We are planning on the identification of other enzymes that are present in the ASD operon, and we will try to do it with respect to acetylseringon and also TCP degradation. Although I personally think that only the first enzyme of the entire degradation pathway is involved in TCP degradation, but let's see, this has to be investigated. However, very likely the acetylseringon degradation continues through logically as it normally is in soil bacteria through the other enzymes of the same operon. And because I showed you at the very beginning that we have we don't only have the one enrichment culture enriched on acetylseringon, we also have other liquid enrichment cultures. So we plan to run similar assays with other liquid enrichment cultures with the possibility of revealing potentially novel enzymes or entire operons. And with that, I would like to thank to all the people that are involved in the OPAC project somehow to a certain extent, or all of our collaborators. And then at the very end, of course, I just realized that I put it there in Czech, although the entire presentation is in English. So sorry about that, but I guess we all understand that the financing came from OPAC project. That is, I would say, the major financing body of your institution, so Institute of Microbiology, but also my group here at UCT Prague. So thank you very much. And